I am Professor Ben Bikman, a biomedical scientist and professor of cell biology. Hopefully that means I know what I'm talking about. Thanks for joining me today. This is a topic that I've been looking forward to discussing, in part because there is so much silly thinking and bad information when it comes to saturated fat, and in particular, its effects on insulin resistance. This is something you've likely heard many influencers speak about, especially those who advocate plant-based diets that encourage you to avoid meat. Often they will cite studies. They will say, well, the saturated fat from meat is causing insulin resistance, so that's why you need to avoid it. I have been looking forward to this topic in part because it's actually one of the areas that I am most authority on, or most authoritative on. Having worked in this field quite substantially through not only my PhD dissertation, but also my postdoctoral fellowship, where I focused on metabolic research for a few years working with Duke Medical School. All right, so with that as the introduction, let's just dive right in. In fact, let's start with some of my own work. Interestingly enough, it's some of this work that is cited by the anti-saturated fat advocates. And so in 2011, I authored a manuscript that was published in a very good journal called the Journal of Clinical Investigation. It's one of the higher impact factor biomedical journals in the field. So very well respected and very difficult to get a paper accepted to publish in that journal. This study focused on the role of a particular receptor that is found on the cell. So on the surface of cells, among the many, many types of receptors or doorways for various molecules to come and knock on, there is something called the TLR4, or the toll-like receptor 4. What we found in this paper published in 2011 is that when TLR4 was activated, it would induce a series of events and steps that would increase inflammation, and that this process would result in the synthesis and accrual of a particular type of lipid called ceramides. Ceramides are the main molecule within a family of lipid or fat called sphingolipids, named after the enigmatic Sphinx, because for so many decades, no one knew what these lipids did. Part of what I helped with was identifying the fact that these lipids do have a metabolic role to play, specifically inducing insulin resistance. So ceramides are a main mediator of insulin resistance, and they do so by blocking the cascade, or the series of events that is downstream of insulin binding its receptor. So when insulin comes and binds its receptor, it would elicit a series of second messenger events, we call it in cell biology. So one thing leading to another, leading to another, leading to another. So I used the word cascade a moment ago, and that's a pretty good word. You can imagine this cascade, this waterfall of events occurring. Ceramides block that up. It basically is damming the cascade up, ultimately causing the cell to become insulin resistant. Now, having said all of this, what is the role of saturated fat here? Well, we identified in this study through a series of experiments that saturated fats uniquely, among the family of all fats, saturated fats uniquely stimulated TLR4, thereby resulting in the accumulation of ceramides and the overall antagonism or the insulin resistance that we're interested in for the sake of this discussion, and was the focus of that manuscript. All right, so let me say that again. So saturated fats were found to be a ligand or a binder or an activator of TLR4. And when TLR4 was activated, it would then create ceramides or induce the synthesis of ceramides and the accumulation of ceramides. And then ceramides would block the insulin cascade, causing insulin resistance. So that was all accurate. It's all true, and it's often cited, my own research, by the anti-saturated fat army. Now, let's take a step out of this work. And I had to do this myself. As I'm telling you this, there's also this sort of undercurrent of my own journey, because I got done that work with this strong conviction that saturated fats really are a problem, This was and we did this work over actually a couple years. It was 2008, 2009 when we were doing this work. It takes so long to get stuff published. And I really was doing this work thinking, boy, saturated fats are such a problem. Now, how did we actually conduct these experiments? This is part of the evolution that I had to go through myself, which I was happy to do because it forced me to change, to look at the experimental model. And by looking at the experimental model more correctly and more lucidly, you're able to challenge the paradigm. So the paradigm here being saturated fat causes insulin resistance via inflammation in TLR4 and ceramides. But how did we actually show that? Well, we did it by directly infusing lipids right into the bloodstream and by directly incubating or treating or bathing cells with these fats. So it was a direct model. We were either directly bathing the cells with the fats or directly infusing it intravenously. Can you see the problem? Is this a physiological model, these experiments, that I'm very proud of? I'm very happy with the work we did, although I very much regret how misinterpreted they've been. And in hindsight, wish perhaps that we would have clarified some of this in the discussion of the manuscript. But is infusing a fat intravenously or bathing cells in a cell culture the same thing with saturated fats? Is that the same thing as eating saturated fat? Of course it's not. Of course not.